Thank you. Thank you, Subis, for the introduction, and thanks for the committee to organizing this presentation, so as we have this opportunity to show our projects. So this project is about the high-dimensional analysis for spectroscopy of exhaled gas from COVID patients. This is a collaborative project with Dr. Subis Basu, and it is funded by the UCF AI and Big Data internal project. So in this project, our focus is on the analysis of the human breath IR spectroscopy. It is motivated by the recent advance in absorption spectroscopy, which allows for the fast and high throughput and highly sensitive spectral intensity progressing of gases. So that includes the human exhales. So we know that human exhales is composed of a mixture of various components. And among these compounds, the concentration level of the volatile organic compounds, which is shortened as VOCs, can serve as a biomarker of various disease, especially in the disease related to the respiratory disease in the airway or lung-related diseases, those similar to COVID-19. And this enables us to do the diagnosis from the exhaled breath of human beings. And this technology, because it is really fast, it allows for real-time diagnosis. So we will look at the spectroscopy data, which contains the fingerprints of the kinds of the compounds. So this project will be approached by synthetic data. So basically speaking, we are going to generate the data by two different methods. One is through the computational simulation, and the other is as a proof of concept. We are going to prepare gas mixtures and then use the spectroscopy, the absorption spectroscopy, to scan the IR, the infrared profiling of from this mixture gas. And then after that, we are going to develop data analysis, which has two targets. One objective is to identify the signature biomarkers. So the first is to estimate the concentration of the biomarkers included in our library. And then we will conduct the multiple comparison and hypothesis test to check if to identify those biomarkers in the library and that are related to the COVID-19. And then the second objective is to directly from the spectroscopy, we want to do a discriminant analysis to discern the labeled and the control data. So currently our approach is focusing on the generating the data. So the idea is that we first collect the compounds. We first record the compounds of various kinds from the human exhaled gas. We have collected the major compounds from human breath, and also we need to borrow the effort, the result from the current publication concerning the COVID-19, which reports the VOCs, the biomarkers, and their concentrations. Also, we also collected those biomarkers of 92 biomarkers of the other diseases related to the airway and the lung diseases. And then we are going to obtain the spectroscopy from a public available database and generate the spectroscopy. That is computationally generated data. The other way is to, based on the concentration and the library, we are going to generate the gas mixture and profile their IR spectroscopy. Use that for the data analysis. So for the impact, so first of all, currently our data is based on the fabricated data. So we will use this as a preliminary result for the proof of concept, and later on, this can be this procedure can be applied to the real data that is collected from human subjects. And the second is we can conduct the real-time detection as well as monitoring the breath with longitudinal type of study. And then this method from the breath can be combined with other techniques to reduce for diagnosis. And also, so after the preliminary study, we will going to detect the subregions, which can further reduce the time and the cost of the absorption spectroscopy scanning in the future monitoring. So as I said, so basically we collected the record from from literature, from the vast literature about first the major VOCs in healthy in the breast of healthy individuals. So we know that there are above 3,500 VOCs that has been observed, but not all of them are observed in the majority of healthy human beings. So those, there are various sources of such VOCs, including the environment and the diet, the drugs, etc. So we just collected 16. We just recorded 16 major VOCs that are reported in the literature, and then we also collected 92 unique VOCs that are reported as biomarkers in the breast associated with the lung and airway related diseases. We also find 3 most very recent literature that use the GCMS, the MS related, the chromatography mass spectroscopy. But those are in these three papers, they reported the result of the analysis result from the mass spectroscopy. Each of them has a small amount of human subjects participating in the study. So less than 100 total summation of the of the control group and the infected group. So the reason is that one reason is that the cost of the gas chromatography process is takes a long time, and it requires a trained researcher to obtain the spectroscopy from the GC process. Comparatively, our proposed absorption spectroscopy, it is much faster. It can be done within 2 minutes. So what we are doing is that we collect those VOCs, the names of the VOCs, and then we construct the infrared spectroscopy instead of the mass spectroscopy, which was reported from the current literature. Also, as noise, we have the names of the other VOCs that are not major VOC reported from the literature. So currently we are doing the data generation by computational simulation. That is a combination of this major VOCs and the VOC reported in the COVID patients. So totally, there are 18 such VOCs together from the three papers. So they are all different. So from the three papers, the reported biomarkers are all different, and we are going to we are going to combine them to incorporate in the spectroscopy. So one group is from the is a major VOC plus noise, which is a minor VOCs in the healthy individual. The other group is the major VOC plus the biomarkers of COVID reported in these three papers, plus this noise as a minor VOCs. So the generation required the concentration, which is also reported from the literature of those VOCs, and also we will combine those reported biomarkers from the other diseases, that the similar diseases of COVID-19. For this simulation. So far we are in the data generation process, and in the remaining task would be the generation of the fabricated gas mixture, and then scanning of those IR spectroscopy from the lab measurement. So we will have this as the preliminary result before constructing the device and collect data in the from real human beings. And then, as we said, we are the big data analysis will be accompanied. This is a complex. This will be complex data. So there will be highly noise. So above 3,000 VOCs detected in the literature. So what we are going to do is we will apply those. We will apply the various type of the multiple comparison methods to compare if each VOC is significant, and adjust for the significance, and adjust the significance to detect those VOCs that are really important from our generated data. So this will be one result, and the other result would be the reduced spectroscopy range of the wave number, which can and then we will detect if the removed spectroscopy is really insignificant, and so we will obtain the reduced range of the wave numbers of the spectroscopy, which can further reduce the scanning cost and the scanning time. Of course, a more direct result would be the diagnosis or the classification directly from the spectroscopy. Instead of estimating the concentration of VOCs, so the way to do it will be directly using the spectroscopy to classify into different groups. So we are going to use various methods, including what the current literature has been accepting, like PCA, the partial least square regression, and the neural networks. We are going to combine various methods, including the tree-based methods, random forest, and we are going to also apply the most recent neural networks in analyzing analyzing the spectroscopy directly, and in identifying the important biomarkers, we are going to use multiple comparison as identification methods and conduct a statistical hypothesis test. So this this preliminary method, preliminary project can be highly impacted very quickly. So one is we will be able one future direction will be the joint diagnosis with the other methods like sound, temperature, etc. And the other is that we can obtain real-time diagnosis of other diseases from the breath analysis. That is the end. Thank you.